All right, so the next two chapters are chapters 12 and 13 are really about how our body defends itself. So we've talked about all these different microbes, right? Hookworms, influenza. We haven't really talked about other alien invaders, but all these microbes that our body is trying to keep out, right? So we've definitely emphasized that some of these microbes are good, we get along, but we have to have a robust system for keeping the bad guys out. And so what we're going to look at in chapter 12 is what we call the innate nonspecific defenses. And then in chapter 13, we'll move on to the acquired specific defenses, which is truly when we get into the immune system. So I try really hard not to use the term immunity or immune system until we get to chapter 13. So what we're going to see here in chapter 12 are defenses essentially that are innate, meaning you're born with them. Things like skin, you come out with skin, right? That's already protecting you from microbial invasion. And they're non-specific defenses, meaning these defenses aren't picky. If it's foreign, we're going to fight it, but we can't really tell the difference between, say, E. coli and salmonella, or one strain of flu from another, but we can often recognize, hey, that's foreign and I should attack it. Okay, and so that's what we're going to be looking at. For those of you who have taken AMP2 already or in there, this is going to look extremely familiar. Sometimes students are like, oh, I like the way you presented it better in that class. Fine, then answer questions that way. I'm presenting more or less the same material. So how do I say this? But I'm not a robot, so it'll be a little different each time. But you know, if I didn't say anything wrong in the other class, you can totally use that information as well. Or you can say, yeah, I know this, and you can ignore this video completely. Okay, so let's go ahead then and jump in here. So as we talk about these body defenses, I did want to point out this is super important. We really need to be able to have our white blood cells, different components of these defense systems, either specific or non-specific, be able to tell self, as in, oh, hey, that's my own liver cell, or oh, that's my own skin cell, or whatever, and not react with it, right? Not attack. And then what we see down here, let me grab the pointer. And then what we see down here is what should happen when there's a foreign cell, right? Immediately we should pick up on the fact that this is foreign and we should attack it. So this idea of having our ability to differentiate self from non-self is super important. And this is an interesting thing. So even our non-specific defenses, this is showing one of the ways that they actually recognize foreign molecules. So they have what's called this PAMP, a pathogen-associated molecular pattern. So these are going to be proteins on the surface of the cell that immediately our immune system is like, oh, wait a second, right? That's a pathogen. And so it goes ahead and destroys it. Okay, so self versus non-self is key to the way these defense systems work. Okay, so again, here in chapter 12, we're in these non-specific defenses, and I'm basically going to split it up kind of like this did, right? So they actually split kind of everything into 3 lines of defenses, right? So this first line of defense is going to involve physical barriers like skin. We'll talk about that. The microbiota, right? So the good bacteria that we harbor, say, on our skin that kind of outcompetes and protects us from other microbes. And then also some chemical defenses, right? Then we'll go into this second line of defense, and that deals much more with things like our macrophages, right? Different phagocytic cells, the inflammatory process, fever, antimicrobial products, etc. So we're really today going to look at these 2 lines of defense, and then we'll come back next week for the third line of defense in specific immunity, right? So really, in our first line of defense, here's where we're at. We're going to talk about skin and mucous membranes, right? So physical barrier, chemical barriers, and then that microbiota that's helping us out. I'll come back to that. Okay, so skin, whoa, I'm hidden. Not that you have to see me. So skin is an excellent barrier in multiple regards, right? So recall the epidermis is made of stratified squamous epithelium. And so the outer layer of skin is all these flat pancake cells squished together, multiple layers of cells. These outer ones are dead. And this is a huge part of our defenses, right? Just that there's so many layers, it would be really hard for an invading pathogen to get through there. Other things that can be really helpful, one, the microbiota, right? So things like we normally have things like Staphylococcus epidermidis living on our skin. And since they like us and they want to live there, they will attempt to compete with any bacteria that are trying to colonize us as well. So that's actually a really nice defense. Maybe we shouldn't be scrubbing with antibacterial soap quite so much. Anyway, other things about the skin. So those outer layers I mentioned are dry, or I mentioned they're dead. They're also really dry, so not necessarily a particularly habitable environment. They're constantly shedding. The technical term is desquamating, right? But those surface cells are falling off. And so any bacteria that manage to stick to the outside of us, it's not going to take too long, right? If they haven't managed to get any deeper, they're just going to fall off. And so that's actually part of this physical defense as well. We'll also notice things like sweat. So this picture is showing some of these sweat glands. The sweat that we exude out onto the surface of our skin, even not like necessarily sensible sweat, like you don't have to be dripping in sweat, right? But we're always actually losing some fluids across the skin. These secretions tend to be slightly acidic, which can be inhospitable to microbes. We produce certain oils that can. That's an interesting one. I was going to say, like fight off bacteria. Some bacteria like them, right? They'll actually use those as fuel, but that's going to keep some microbes as well. Sweat can rinse our skin off, etc. Oh, salty. That was the other thing I was thinking. I was like low pH, right? It's slightly acidic, and it can also be salty, sweat, right? So those would be some of those chemical defenses. Now, of course, not all of us is covered in skin, right? Sometimes we have mucous membranes. So this mucous membrane is going to be what's coating all the openings into the body. It actually looks really similar. So it's going to be stratified squamous epithelium, but it's not quite as tough. So it's actually not keratinized. It's not filled with this really tough protein called keratin. And so that makes mucous membranes a little more vulnerable to microbial attack. So we have other tricks to defend ourselves. So something as simple as this little girl's tears. Isn't that cute? Right? I feel bad, right? These tears, they're cleaning the surface of the eye, right? So some of these mucous membranes have ways to flush microbes off the surface. Those tears also contain an enzyme called lysozyme, and lysozyme helps digest bacteria. It particularly digests peptidoglycan. So lysozyme in tears is meant to protect our eyes from bacteria. We see mucous membranes having all sorts of mucus, right? So this sticky substance, that's going to help trap microbes, right? So protecting maybe deeper structures in our airways, for example. And then, of course, in our nasal cavity and also in the trachea, we have ciliated epithelium. So these cilia are actually beating and propelling that mucus up and out of the airways. They call that the mucus elevator. Isn't that lovely? And if you think about it, it's actually kind of ingenious. So say your trachea, for example, it's a way to trap microbes, dust, anything that's coming in through your trachea and then bring it back out, right? And so you know how sometimes you kind of have to just like cough a little, you're pulling that mucus up, and then I don't know if you notice, you immediately swallow it, right? And that's actually brilliant because then it's going to your stomach, which is filled with hydrochloric acid, and we just like destroy the bacteria there. So kind of a maybe a weird reason that the respiratory and the digestive tract are so closely paired, right? Sharing some of these structures. Anyway, what else was I going to mention here? Oh, saliva. So we see saliva again. That's kind of bathing, clearing the surface of the mucous membranes in the oral cavity. The vagina would be a mucous membrane. We already mentioned how we have that association with lactobacillus colonizing there to prevent things like yeast infections. If you think about the urinary tract, right, the urethra, as that exits the body, that's going to be a mucous membrane, but urine is constantly flushing through there to pull any microbes out. It's very acidic, right? So all of these things, all these extra enzymes, acidities, secretions, etc., on mucous membranes is because they are a little bit more vulnerable, right? But with both of these, right, skin and mucous membranes, as long as they are intact, right, undamaged, they provide a really great first line of defense for us. There's the bacterium being trapped in the cilia, getting carried out by the mucus elevator. And again, just a little summary there of all these different things. Sweat, low pH on your skin, acid in your stomach, and on, right? Nice little review there. Okay, so then our second line of defense, it's still nonspecific, it's still innate, but we have a lot of stuff going on here. So inflammation, right? Inflammation is a very standard body process in response to invading microorganisms. In fact, just in response to injury, because we assume that there's probably some microorganisms involved or willing to take advantage of said injury. So I like this picture just because it's a good reminder of the signs and symptoms of inflammation. So in the original Latin, your signs and symptoms are rubor, which is redness, calor, which is heat, tumor, which is swelling, and dolor, which is pain, right? And that leads to a loss of function. So kind of fun, rubor, calor, tumor, dolor, if you want to remember signs and symptoms of inflammation. And if we look at the actual process of inflammation, right, those signs and symptoms should make sense. So let's run through this real quick. So again, inflammation is in response to injury. So our classic example here, we are stepping, looks like on a rusty, well, I don't even know if it's rusty, on a nail, right? Bacteria coming in with it. Now, notice one of the first things that happens, we have this immediate, I'm going to switch to the pen here, we have this immediate vasoconstriction. Because the blood vessel is worried about bleeding out, right? It's actually going to constrict itself, try to slow down, cut off blood supply to allow hemostasis, right? To allow clotting. But then the really characteristic parts of inflammation take over, right? So the cells, did they show them? Mast cells in the area, these are related to basophils. So mast cells release, they say chemical meat eaters, they release histamine, right? So histamine is going to be a vasodilator. So this vasoconstriction is very short-lived, right? We'll move into vasodilation, and the vessels, as they're relaxing, right, getting bigger, this is going to increase blood flow. That's why it's becoming warm and red. And it also becomes kind of leaky. So the capillaries here, they're made out of simple squamous epithelium. And so the junctions between cells are kind of easing up to allow this dilation. And so that makes them leakier as well. And so here we see things like white blood cells coming to the site of injury. So that's a great part about this leakiness. And then we also see plasma exiting. And so this is why you're going to get things like edema. And then the swollen tissue, the edema starts pushing on nerves, and that's why you get the pain, right? So all these things start to make sense, but it's really important to realize, right, that the mast cells release histamine, and that leads to vasodilation and leakiness of these vessels. And so then what we're seeing here, right, all that edema, right, the plasma starts kind of filling up the area. There's talk that actually helps kind of wall off the site of infection, right? Kind of gives it boundaries so that bacteria and stuff aren't moving beyond that. Notice that they're pointing out here pus. So pus is going to be all that plasma, dead bacteria, dead white blood cells, right? And that is an important part of the healing process, right? Kind of a side effect of all those white blood cells and things being able to move out. So we'll talk more about those white blood cells because particularly their macrophages and neutrophils are going to be the main ones coming in, and they're actually sensing chemicals that are being released at the site of injury, and they are actively coming to the site and looking to help out. So same thing, this is table 12.3 out of your book. It just has way too many words to show in a PowerPoint, but it is a nice summary of what's going on there. So, and this is, I just think this is so cool. So look at these neutrophils. They want to get to the site of infection, right? They've caught the smell, some chemical mediator, and so they're using chemotaxis, if you remember that term. They're moving towards a chemical stimulus. So they're being carried freely in the blood, but then as they sense that they're approaching this site of injury, right, they actually move themselves over to the wall of the capillary, right? Things flow fastest down the center of a vessel. And so they actually move to the side, stick themselves to the endothelium and kind of start doing this like army crawl along the edge of the blood vessel until they can find one of those little openings. And then they use this, it's a process called diapedesis, but they're basically using this amoeboid movement, and they can squeeze themselves out of the blood vessel and to the site of injury. And I just love that they're so enthusiastic about this, right? They're not running to hide. They're coming to help. Okay, so the other thing I did want to point out, this picture looks crazy. This is from a previous text, but what I wanted to point out, right, is that so what we're looking at here was an injury, right? So some kind of trauma, but this could also be infection. They're pointing out cancer, etc. Your body has a bunch of different chemical signals. Wow, this picture is really kind of messed up. And what I wanted to point out, we don't need to know all of these, right? But some of these chemical signals are vasoactive. So we were just talking, for example, about histamine, right? And how that leads to vasodilation and that increased permeability, right? Other chemicals that are released from the site of injury are going to be what they're calling chemotactic chemicals. And these are those chemicals that would actually be attracting the white blood cells, right? So endotoxin, that could actually be like a toxin on the bacterial surface. What's another good example? Those PAMPs that we mentioned earlier, the pathogen, they're always associated with a pathogen. And so that's going to be a signal for white blood cells to come as well. And then we'll talk about some of these other chemicals that maybe fill a role in both, right? But think about at our site of injury, because we want to stop bleeding, there's always going to be platelets coming. And those platelets then can also signal phagocytic cells to come to the site as well. So lots of different chemicals. They keep finding more and more of these little chemical signals. So this list will keep growing. Okay, so we talked a little bit about inflammation there. Let's look at the phagocytic cells that were coming to help. Again, table 12.2, really nice, really way too small to use in a presentation. But you could check this one out. It's talking you through the actual process of phagocytosis. So a couple things, I guess I can still just kind of walk through here. You can see this in your book as well. So these white blood cells are using chemotaxis, right? They're actually attracted to the site of injury or where they're catching the scent of a microbe, right? They need to actually be able to attach to the foreign cell, right? Or get the foreign cell to attach to it. And so they have special receptors on their surface that do align with those PAMPs. And so they can very easily kind of pick up and stick to bacteria. So then we're going to go ahead. Phagocytosis is truly like the engulfment of this microbe. And so here what we see happening, right, is it kind of just an impocketing of that cell membrane with lots of bacteria already stuck on receptors. And we're going to go ahead and bring that inside of the cell. It's going into, oh, they lined these up. Super cool. So 3 up here says engulfment, 3 down here says engulfment. 4 is pointing out that we're putting those bacteria in what's called a phagosome. It says just a little vesicle, a little bud of the cell membrane, and then we are going to attach it to a lysosome. So lysosome, the poor organelle that no one ever pays attention to in Gen Bio, right? It's this little pocket of enzymes that are being made by the cell. So over here, they're actually showing the production from the Ruffyard to the Golgi apparatus, the production of these lysosomes, these little packets of enzymes. And we're going to fuse that to the phagosome. And so we get a phagolysosome, which is a mouthful. But basically, this is going to let us put those enzymes in with and kill and destroy the bacteria. We can then go ahead and dump what we don't want out of the cell. We could also use some of the nutrients, some of the building blocks of that bacteria, but we also have the ability to just go ahead and dump that. So this is a really nice kind of run through of the details of phagocytosis. So the main cells we're looking at here are neutrophils and macrophages. Now, they act a little differently. So neutrophils, notice this is like 60 to 70% of your white blood cells. This is like by far the most common white blood cell. But I think of them as the little kamikaze pilots of this setup because they can really only ingest maybe like a dozen bacteria before all of the enzymes and the lysosomes and the destruction becomes too much, and the neutrophils actually self-destruct as well. So when you think about pus and there being dead bacteria and dead white blood cells, a lot of that is going to be neutrophils. But fortunately, they are quite abundant, right? So that might not be too big of a deal. That is different than the way macrophages work, even though they're both doing phagocytosis. So the macrophages, literally macro, large, phagocytosis was like cell eating. So literally big eater. These are very large white blood cells, and they can just keep going and going. This is one of my favorite pictures in biology. This big cell back here is a white blood cell, a macrophage, and he is extending this little process. Talk about chemotaxis, right? He can smell, he can sense this little bacteria. You might not have even seen him down here. And he's just reaching out and he's going to grab him. I think that's so cool. And so they can just keep doing this. They do not self-destruct. They just keep consuming. We've talked about this a little bit, right? So some of these macrophages are floating. So when they're in the blood, we would call them a monocyte, and then they can move out, right, into the tissues, travel through the lymph, back into circulation. When they're in the tissue, we call them a macrophage. We also have some, they come from the same lineage, right? They're monocytes, but then end up kind of staying put, right? So we will see some macrophages that basically just hang out in the spleen, right? And more or less stay put there in the lymph nodes and kind of stay put there. We have some specialized ones like the Kupffer cells in the liver or the dendritic cells in the skin, and those also stay put. So this is another cell that I wanted to mention here. It's a natural killer cell. Now note, it is not a phagocytic cell, but there's nowhere else better to talk about them. And it's the same idea. It's part of these innate nonspecific defenses. And we usually talk about natural killer cells being the immunological surveillance. So These are funny little guys. They're actually much more closely related to lymphocytes, and we'll look at like the family tree of blood cells. These are not closely related to macrophages or neutrophils, but they're working on this innate nonspecific side. And so they're not phagocytic, but they're still destroying the foreign pathogen. So the end result is the same. So this is our natural killer cell. And here it's identified a cell. One of the things they're actually really good with is destroying virally infected cells, right? So say COVID-19 got into one of your respiratory cells. If a natural killer cell sees that, it can destroy it. And they also tend to be kind of the early warning system on cancer as well. So they'll pick up on cells that have a mutation in the DNA and often take them out as well. And so, here we see the natural killer cell kind of just scooting up on next to them and then just opening fire, right? Shooting a bunch of holes. They use a chemical called a perforin to perforate that cell, and then it's going to actually die due to the osmotic stress, due to water rushing into the cell and making it explode. So not phagocytic, but this is the best place I could think to talk about them. Okay, so again, in the second line of defense, inflammation, some phagocytic cells, and then all sorts of different chemicals. And we're just going to talk about a few of these. Okay, so sorry, this is a little silly, but what we're going to see is that we have a variety of different kind of antimicrobial chemicals. So some of them are working, like we saw, those chemotatic factors that actually like draw macrophages and neutrophils to the site of infection. There's this picture of a fence is my vision of a molecule called an interferon. And so this is a molecule that a virally infected cell releases to warn the neighbors to put up their defenses. So interferons tell the neighboring cells to set up their defenses. Hey, I caught the virus. I'm going to die. You do what you can to save yourself, right? That kind of thing. We'll also see that some of these chemicals can just act as tags. So they may not have the capability of destroying the cell themselves, but might make it much easier, say, for something like a macrophage to actually notice, right, and identify this as foreign and destroy it. And then this one down here is kind of fun. So this is what's referred to as complement. It's a series of proteins. I want to say it's 11 proteins. They're all just circulating in the blood. But then what ends up happening is when we find a foreign cell, and often the way it's going to recognize this foreign cell is if it has been tagged by an antibody. So it's called complement because it complements the activity of antibodies. But so what these proteins do is they embed themselves into the cell membrane, and you see this kind of ring that they're assembling. So it just builds a pore. So it's called a membrane attack complex, right, or MAC, that the complement proteins are making. And so again, this is going to become osmotically fragile and explode. Okay, this is a better picture than my fence picture for interferons. So here we see, right, this is the virus. It's infecting our cell. The cell, this is so funny, right? It can't stop the virus from taking over, right, and copying and making new viruses that are going to be released. But it sends out this Hail Mary, right, of like, save yourselves. And so we see interferons moving over to a nearby cell. And the idea here, notice he's not getting in. So the idea would be like this cell takes down like the receptor or something that virus needs in order to infect them. So here we see some of the cells may be able to, with the early warning system, be able to degrade the virus, block the replication, make different antiviral proteins, etc. So anyway, I think that one's pretty cool, interferon. This is a close-up of the complement system. And so again, we've got this whole series of different proteins they're trying to show in different colors, but we're building this ring. And again, that's going to punch holes in the cell and it will explode. Okay, so those are the big parts. Oh, fever. I don't have fever in here. Fever is really important too. It kind of, I should probably talk about it with inflammation, right? It kind of becomes that body's systemic response um, to something like an infection. And so if if you're running into inflammation and we're worried about a more systemic infection, the hypothalamus is the part of the brain that regulates body temperature, and so you can send it signals to go ahead and up the set point, right? To turn up the thermostat to up the body temperature. And again, just a reminder, that's not necessarily going to just fry these microbes, but if we can push them just out of their optimum growth curve, right? Remember, right? Everybody has an optimum temperature. If you can scooch that temperature just enough to force them to replicate more slowly, that's beneficial. And it does seem like those higher temperatures also may help our cells, right? Like the white blood cells actually metabolize faster. So yeah, sorry, fever should be in there as well. Okay, so then this list is so that I don't forget to talk about these things. Because, how do I want to say this? When you think about these innate defenses, they're kind of spread throughout the body, right? These macrophages are everywhere. Complement is just circulating and on and on. And so this is kind of a chance to point out some of these systems where you're going to see these particularly richly embedded with these cells or particularly involved in these processes. So blood and the lymphatic system, we should all be familiar with. We'll talk about them some more. The reticular endothelial system and the mononuclear phagocyte system, you're not going to see those listed as systems in an anatomy text. And so I think I have the tendency to kind of be like, it's not really a system, but it is really important to think about how, I would say, to me, the biggest thing about reticular endothelial system and mononuclear phagocyte system is we kind of see them popping up in this connection between the blood and the lymphatic system. Let's take a look. So what they're trying to show here is this reticuloendothelial system. So notice what they're showing is that between cells throughout your body, right, you have connective tissue. So they're showing, if you remember connective tissue, it's made out of things like collagen fibers, elastic fibers, and reticular fibers. And so these reticular fibers tend to be much more kind of branchy and be willing to kind of act as like a spider web and kind of hold on to some of these cells. So some areas like the spleen and the liver would be super rich in these reticular fibers, and that would actually help hold on to and support some of these white blood cells kind of hanging out there. So that's maybe like the reticular part, and then the mononuclear phagocyte system, the other thing to keep in mind is, I think what they're saying, and you could actually show this in either picture, right, are phagocytes here, macrophages, neutrophils, dendritic cells, or a specialized monocyte. Again, they're kind of embedded throughout this connective tissue throughout the body. So those two systems, I think, really go together. Reticuloendothelial mononuclear phagocyte system, the phagocytes kind of inhabit that reticuloendothelial, the connective tissues of the body. So then when we look at it, remember the connection between blood and the lymphatic system, right? So stuff is always leaking out of the capillaries. So we see these arrows going out, some stuff gets back in, but we have all these fluids and nutrients and hormones and cells sometimes leaking out of the capillaries and moving into the extracellular fluid. And then it's often the lymph, they're showing that kind of here in gold, right, that's going to pick up the extracellular fluid and return it to the cardiovascular system. So this picture, right, blood, substances are moving from the blood to the extracellular fluid, and they're pointing out, hey, a lot of that extracellular fluid, as you're bathing, you're really bathing what we're calling that reticuloendothelial system, all these connective tissues. And oh, by the way, that's where we've left this rich supply of white blood cells. So if something comes out of this capillary, we're there to grab onto it, right, and engulf it and destroy it. And then The lymph, right, as those fluids move back into the lymphatic system, they're not showing it on this picture, but before it goes back to the heart, we're going to run it through a filtration system in the lymphatic system. So I think they're just trying to show how tightly all these things are tied, right? Those fluids are going back into the blood. And the blood's super important, right? Because it's propelling, it's carrying all of these white blood cells around the body, so they can move in and out of tissues and protect us. So anyway, those four are kind of all connected here. Okay, so then I believe the last thing that I want to talk about, is hematopoiesis, kind of the production of blood, in particular because we're interested in a lot of different white blood cells in these body defenses. So I'm going to switch to this picture and just kind of give us a run through of what's going on. So here we see all the different adult white blood cells, right? So red blood cells, platelets, neutrophils, basophils, eosinophils, monocytes, which become macrophages, and then we'll get into lymphocytes more next week, right? Our T lymphocytes and our B lymphocytes. Oh, and there's their cousin, the natural killer cell, right? So we have a lot of diversity in our blood cells, but they are all going to come from the same stem cell. So we have what's called a hematopoietic stem cell, and this is located in the bone marrow. And really early on, right, so remember stem cells are just cells that are able to keep dividing and dividing and dividing. And we're going to split really early on then into two different lineages of stem cells. So this group over here, it's kind of hard to see in this picture, it's called a myeloid stem cell. And then these cells are coming from a different lineage called a lymphoid stem cell. So they're like cousins, right? But we're going to see different cells come out of these two lineages. The picture in your book didn't go into that detail. So I pulled this image from a previous text. So the myeloid stem cells are going to make most of our cells, right? So from the myeloid lineage, we get red blood cells, we get platelets, which notice they're not truly a cell. They're fragments of a cell called a megakaryocyte. In our white blood cells, right, so they've put a box here around all of our white blood cells. From the myeloid lineage, we get our neutrophils, remember our little kamikaze pilots, our basophils, which release histamine. Notice they're pointing out down here a mast cell. So that's going to be basically a tissue-embedded basophil. Slightly different, but for us, they both release histamine. And then eosinophils, these tend to go after like larger pathogens, like multicellular parasites, like a tapeworm or giardia, something like that. And so eosinophils are another white blood cell that we didn't mention yet. And then, of course, I mentioned the monocytes, and those can have lots of different names depending where they are, macrophages and dendritic cells, etc. Right? The lymphoid stem cells just make T cells, B cells, and NK cells. Okay. So some of the stuff that you're going to come across, right, is when you do for like a patient, if you've done a complete blood cell count, you actually get percentages of each of these different white blood cells. And so you can tell whether this is normal or not, right? So we mentioned with the neutrophils, those should be something like 60 to 70% of the white blood cells. You would notice, right, if that's off. If you saw someone who had huge numbers of eosinophils, because eosinophils are actually really rare, they're like just a couple percent of your white blood cells should be eosinophils, then you start to say, oh, this person maybe has a parasite. You'll also see higher levels in some allergies. Basophils, they should be less than 1% of your white blood cells. And so again, if you see higher levels there, you might be concerned about allergies or how much histamine would be released. So knowing kind of those percentages, you can tell something about infection. And of course, if you have oh, I guess leukocytosis, an increased number of all of your white blood cells, right? So all your white blood cells together are leukocytes. That would be indicative that there's an infection taking place as well. And so that can be really useful information. So then I guess I didn't want to end with hematopoiesis, the production of blood. The lymphatic system is a real system that we have a whole chapter on in anatomy that's really has a really important role to play here as well. So we saw that the lymphatic system was kind of returning all those fluids to the cardiovascular system. And this picture is nice. It shows kind of how widespread this is throughout the body. We'll look at a little more detail here, but these places where we have little lymph nodes, right? So here's a lymph node. And so what we have are these lymphatic vessels bringing all the fluids that they've collected out of this capillary bed. And they're going to bring it through the lymph node, which essentially acts as a filter. So there's tons of macrophages, tons of lymphocytes, and we're going to give this basically ends up being a chance to do surveillance on the fluids of the body, kind of get an early alert if there's a pathogen in the body, because we can activate lymphocytes and different phagocytic cells, start antibody production, all that kind of stuff. And so these lymph nodes are super important there. Oh, so that was my other one, sorry. In the lymphatic system, other things to notice, the thymus is an important lymphatic organ. That's actually where T cells are going to mature. So that kind of becomes like the nursery for T cells. The spleen is a really important lymphatic organ. Again, it kind of is an area to filter. This time we're not filtering lymph, but we're filtering blood. We get rid of old red blood cells here. We have a ton of macrophages in the spleen looking for pathogens. MALT. So MALT is mucosal associated lymphoid tissue. And so you'll find this anywhere. Here's another example, the tonsils, right? Mucosal associated lymphoid tissue. This is going to be more like diffuse. It's not truly like an organ, but it's diffuse collection of lymphatic tissue. And again, richly embedded with macrophages. A special example of MALT is GALT, gut-associated lymphoid tissue. Particularly, we call them Peyer's patches in the small intestine. Think about how many bacteria are in the small intestine, and so we have this protection to keep them from getting into the rest of the body. Actually, that's the appendix as well. Seems to have a role here in the lymphatic system. It's richly, if you look histologically at the tissue of the appendix, it has a very rich supply of white blood cells. So probably playing some sort of defensive role. Okay, so next week we'll move on to chapter 13 and get into the specific defenses. And honestly, this stuff is cool, right? Like lysozyme, we break down peptidoglycan, we can do that. But next week, these specific defenses, the true immune system, to me, that's when stuff starts to get super cool. So we will see you in lab.